So on the first slide, you can see a 3D model of an enzyme. And what you can see is that it is basically a protein. And the protein, as you probably know, is created of amino acids, and they are linked together with peptide bonds. And here is as a review as a diagram of how a protein is being formed. So when in the DNA there are the bases A, T, G, and C, and they are there to encode basically the how proteins are being built. So when there is transcription and translation, the the DNA is being read, and then based on the code in the DNA, you can make proteins, so chains of amino acids. And the primary structure is just in which order these amino acids are put together. And based on that order, there can be a secondary structure, which can be an alpha helix, or there are also beta sheets that are more of like folded chains of amino acids. And then then there's the tertiary structure, and this structure is kind of the three-dimensional form of a protein. And that's also in some enzymes, especially digestive enzymes, the structure that they have. So there's just one protein, and when it is an enzyme, it has a catalytic effect on a biochemical reaction. But there are also much more complex proteins and also more complex enzymes, and that's where the quaternary structure comes into play. This is basically when you have multiple tertiary structures, so multiple proteins bonded together to form a complex. So this is the case in larger enzymes as well as other proteins that are linked together to have a joint function. And you probably know that enzymes are just another form of catalysts, and they are biological catalysts, so they are present in every living organism, and they speed up chemical reactions. And you probably know that most proteins denature when it gets too hot. But from chemistry class, you probably also know that most chemical reactions don't take place until there's a certain temperature or a certain amount of kinetic energy around them. And this is why some reactions couldn't occur in the human body if without catalysts. So let's say you want to break down food or something like that. You have to do that. You have to do that with an enzyme because otherwise you'd have to increase the temperature um like to an amount where the chemical reaction would occur spontaneously, but the problem then is that the body temperature would get way too hot, and the proteins in your body and also other structures would get damaged, and then you'd eventually die. And so enzymes are just the biological form of catalyst, so they reduce the activation energy. And this happens when a substrate molecule binds to the active site of an enzyme. As you can see, here is the substrate molecule, and then when and then the enzyme breaks, for example, bonds between inside the substrate molecule to create two distinct product molecules. They are represented here in two different colors, and then once the reaction is completed, they uh, they get apart and they can leave the active site of the enzyme again. And so the enzyme is free for another substrate molecule to bind to its active site and um, catalyze another reaction. And this is very important, as I've already said, for human life or life in general to be able to exist. Okay. Here is another diagram of inhibitors. So inhibitors are basically molecules blocking the active site of the enzyme. This is the case with competitive inhibitors. Yeah, this is the case with competitive inhibitors. They bind to the active site, but they can't be broken down, for example. And that is a problem because the real substrate molecules then cannot enter the active site and cannot be made into products. And this is why the chemical reaction is slowed down by competitive inhibitors. There are also non-competitive inhibitors, but these are molecules which bind to the active site, to an allosteric site, which is not the active site, and this causes the enzyme's active site to to get another form. As you can see here, there are there the real substrate molecule can't bind anymore, and so the enzyme activity is also inhibited. Here is a diagram of how a chemical reaction usually works. You have the reactants and the products, and in between there's a change in the amount of energy of these molecules. For example, in this reaction, the reactants have a much lower amount of energy inside them than the products, and therefore there are we need to like increase. This reaction won't happen spontaneously, as there is a lot of activation energy, as you can see here, to bring the reactants to the higher level that the products have. And this is an example of an exothermic reaction, or one where the reactants are at a higher energy level than the products, and their energy is being released. But even in this type of energy of reaction, there is activation energy, although it is much lower than in endothermic reactions. And even here, there may be a need for a catalyst. And now we can compare the energy diagrams of catalyzed reactions without catalyzed reactions. As you can see, the curve above, the purple curve or pink curve is this curve has a much higher activation energy, as it is the uncatalyzed reaction. The catalyzed reaction has a much lower activation energy, and this is exactly where enzymes come into play. They reduce the activation energy and therefore speed up biochemical reactions. You may have heard of the Michaelis Menten theory, which is basically it is concerned about the rate of an enzyme catalyzed reaction, as you can see here at the top, there's Vmax, or the maximal speed of the reaction rate. And this is basically if you have the active site, which is being blocked by a substrate molecule, and then you have and then it is being released again when the products are formed, how long it takes for the reaction to occur, and so how long it takes for the active site being like blocked by a substrate molecule. This is what defines how fast an enzyme catalyzed reaction can be. And there's also another value, which is Km, or half the full full speed of the reaction, and how long it takes until that is reached. And this is actually quite important, as when Km is very small, this means that that the enzyme has a has a good affinity for the substrate molecule, so the reaction starts really fast. However, then the the actual speed of the reaction doesn't really change that much. Whereas when it takes a long time for half of the um, maximal speed to be reached, the reaction starts very slow, but it can still reach its maximum speed when there is enough substrate present. And in this equation, you see you have the enzyme, the substrate, and the enzyme and the substrate together, they form an enzyme-substrate complex. And after that, we have the product and the enzyme. And as every other catalyst, an enzyme forms an intermediate product, which is like on the top of the activation energy. There is an intermediate complex, and afterwards, the enzyme is being released as it was before the reaction, so it isn't consumed during the reaction. Here are two major influences of on enzyme activity. As you can see here, here's the temperature. As you know, chemical reactions are usually faster with rising temperature, and you can see that here until this point. But as enzymes are proteins denature at a specific temperature. So the hotter it gets, it will eventually get to a point which is usually 40 degrees Celsius. There, the reaction rate will decrease dramatically because the enzymes are being destroyed by the heat. And this is actually the reason why it is so dangerous to have fever that is very, when your body temperature is way too high, this can lead to damage in proteins and tissue. That's why fever temperatures which are way too high can be deadly. Also, another influencing factor is how acidic, or the environment in which the enzyme functions is. Here, the red curve is pepsin, which is a molecule, an enzyme present in the stomach. And as you all know, the stomach is really acidic, and this causes this causes the this this necessitates that the pepsin enzyme is made in a way that makes it very very active at very low pH. Whereas most other enzymes, for example, amylase, which is an also a digestive enzyme which is present in your mouth, in your saliva, that is more like in a neutral pH range of about 7, very active. So the enzymes have to be adapted to where in the body they work. There are also other factors which influence the activity of enzymes, like um, inhibitors, and there are also feedback loops. For example, if you have a chain of multiple enzyme complexes which are forming a product, you only want to form the product when it's needed. And therefore, for example, when there is enough product produced, the product can then go to the beginning of this whole chain and then bind to an allosteric site and act as a non-competitive inhibitor, which is called feedback inhibition. So if there's enough product, you don't really need to produce any new one, and so the products directly act as inhibitors. And also how the salinity is also something that affects enzyme activity. Now we'll be looking at an example of a real enzyme. You may have heard of stomach ulcers and the bacteria which is called Helicobacter pylori, which causes them. So Helicobacter pylori, or just H. pylori, is a bacteria that causes infections in the stomach. And that can be really, I hope no one has experienced it. I personally don't, but I've heard it's really like not you don't feel good at all if you have this sort of infection. And why in the stomach, you may ask? Because probably if you've learned about the digestive system in school, you have learned that the reason why there's such a low pH in the stomach is not just to break down proteins, although this is also an important function of the low pH in the stomach. The high acidity in the stomach also causes bacteria, viruses, parasites, and other invaders to be killed. So they can cause infections, for example, in the intestines. And so it's like disinfecting the food and everything you take up through eating or drinking, so you don't get sick. But H. pylori uses a reaction with the enzyme urease, which basically breaks down urea to um, ammonium, and this is, as you can see here, the HCO3 minus, OH minus, and NH3 are all basic molecules with a high pH. So around the bacteria, when it breaks down urea to these product molecules, it has sort of a shield that protects it from the acidic environment it is in, and therefore it can cause these infections inside the stomach, even though the pH is very low there normally. But as it can change the pH with this enzyme, the infection is possible. Now we'll look at an experiment that you could do maybe in school if you have maybe you have done something similar when you talked about enzymes in school, or you could ask maybe your teachers to do this experiment if you have time and they allow it. So the materials you need are a conductivity meter to test how much how much electricity flows through through a solution. And this is the case when there are a lot of ions and present in the solution, and you want to test that. And there's a magnetic stirrer with a stirring bar. This is being used so that like everything is homogeneous in the solution. Then a beaker, 100 milliliters, a measuring cylinder, a pipette, and then you need urea solution, which is like the reactant, and urease solution, which is the enzyme, and copper sulfate solution, which is an inhibitor. It is a competitive inhibitor, so it blocks the active site of the enzyme, and we'll see what happens then. So the first step in this experiment is to transfer 50 milliliters of 5% urea solution into a beaker. So you transfer the urea into the beaker, and then you have to place the measuring probe inside the beaker, and then you have to stir the whole solution. As you can see here, we have to make it homogeneous. And then you add the copper sulfate in one of the probe. So once you add it, the ability of urease, so the enzyme, is inhibited, so the reaction can't take place, or not as much as it would without the copper sulfate. Oh, wait. Here you see a graph that you'd see if you do this measurement for about 8 minutes. And now we'll look at the two different diagrams together. So here you have the diagram with copper sulfate added, and the one without copper sulfate. And as you can see here, as long as you don't add copper sulfate, which happens about here, the amount of product is getting large. More and more product is being formed. So ions, which you can measure with a conductivity meter, and then once you add copper sulfate, which is an ion itself, which causes a very fast increase in conductivity, then afterwards the reaction doesn't really continue. It is being blocked. The amount of product in the solution stays about the same. And this is the case because copper sulfate is a competitive inhibitor, so it blocks the enzyme, and the enzyme can't react. And then in the experiment without copper sulfate, you first see also an increase in the amount of product being formed, but it doesn't really stop. So it continues until no substrate is left. And here this experiment shows the effects of inhibitors. And now we are you will be looking at an example. I think we can try making breakout rooms. I have to see if I'm able to do that. We'll see if that works. But I send you a document in the chat if that works. And then you have an activity that you can work on. Okay, wait. I think it doesn't work with the document. So we may have to do it together. Okay, yeah, maybe I can email it. Maybe 1 second. And yeah, okay. I've sent an email to everyone who has registered, and the people who haven't filled out the Google form that we sent out, um, they they can just write the email address in this chat, and I can send the document to them too. Um, could you reply, write in the chat if you have received the document? Okay, perfect. Okay, wait. Um, I'll try to share my screen so we can discuss the activity together. Okay, so I hope everyone has received the PDF, and I hope you can see my screen now. So for the so I'll divide you into breakout rooms, if the technology works. I am, but first I'll explain what you have to do. Here is the description an um of an experiment. The first one is on the influence of molecules with similar structures on enzyme activity. So what happens if there is not just a substrate molecule present in a solution, but also one that has a very similar structure to the substrate molecule? Will that affect the reaction rate of an enzyme catalyzed reaction? You have the instruction, how the experiment works, and then at the end here, you have to interpret the results. For example, here is a color change and here not, and then you have to um, explain the results with the different molecular structures of the substrate molecule and the similar molecule. And the second activity is on the influence of heavy metals on enzyme activity. So heavy metals, these can these act like toxins for enzymes, and the enzyme doesn't work anymore after it. And you can see what happens if you add um, copper sulfate, the toxin, how it will affect the reaction. Um, Here you just have from left to right. These are the same uh, uh, re reaction tubes, but just with different numbers. So 1 is 4, 2 is 5, and 3 is 6. And what also happens if you add EDTA, which is a chelator that can neutralize the um, yeah, the copper sulfate molecules. Okay, I'll try to wait. Um, okay, I'll try to create breakout rooms. So, can you enter the breakout rooms? Has it worked? Okay. Okay. Did it work now? Okay. Um, I think we have to do it without breakout rooms. Um, but yeah, then we do it just all together. I think that maybe works better. So for the first activity, I'll try to share my screen again, which is about the influence of molecules with similar structures on enzyme activity. And this experiment is pretty much the same as the one I showed you in the presentation. You also need reaction tubes, pipettes, urea, which is the substrate, an indicator solution, which causes a color change, the urease solution, and the thiourea solution, which is has a similar structure to urea, but isn't can't be like um um there isn't a reaction that can happen inside the urease enzyme. And now in the first test tube, you put just the urea solution without urea solution, water, the indicator, and the enzyme. And as you can see here, there is a color change. And now, um, could you write in the chat what like what you think is the reason for um yeah, for the color change in the first test tube? Exactly. It is, in fact, the reaction between the enzyme and urea, which produce the product, which cause the color change, which can be indicated with the phenolphthalein indicator. This is an indicator which changes its color with a changing pH inside the solution. Yeah, this is a perfect answer that you can see in the chat. Okay, amazing. Now let's look at the second test tube. As you can see, there is urea solution present, the same amount as in the first test tube. and then as there's thiourea solution present, no deionized water instead of the instead of the wait instead of water we have thiourea solution, and again indicator and enzyme the same as in the first one. And now look at the color change. Do you see a difference between the first and the second one? And do you have a guess why this is the case when looking at the different molecular structure of thiourea and urea? You can write your answers in the chat. This is a perfect answer. Yes, in the second test tube, there are competing substrates for the enzyme's active site. And one, as you can see here, the urea, this is the actual substrate, which you want to form the products out of, like ammonia and stuff like that. And there's urea, which has pretty much the same structure. The only difference is instead of an oxygen atom, it has a sulfur atom attached with a double bond to the molecule. And therefore, they both have the same shape. And so with the lock and key theory, can they both enter the enzyme's active site, but thiourea can't be like there's no change in thiourea. It just blocks the active site, no product is being formed, and therefore urea can't be catalyzed. And this is the and now a question for you. You can also write in the chat, or if you think that what happens if you now we still have the case there's urea and urea, they are competing for urease's active site. What happens if you increase the amount of urea in the solution but keep the amount of thiourea the same? You can also write your answers in the chat. Yeah, exactly. So if there will be a higher rate of the reaction because the concentration of urea is higher in comparison to thiourea, and therefore it's a higher probability that the actual substrate meets with the active site. And that's a perfect answer. Um, I just figured out how to share the PDF in the chat, so you can open it here now too if you want to. Let's look at the third test tube. Here you see no color change. There's only telluria present. What tells us that? Uh, what does this tell us about what happens to telluria inside the enzyme? Exactly. So the enzyme can't do anything with it as it is not the real substrate. Perfect. So, now that you understand that, we can move on to the second example, which is the influence of a heavy metal like copper sulfate on enzyme activity. As you can see, that it is again pretty similar. Now it is a bit more complicated with a few more steps. So you need EDTA solution, which kind of it can neutralize the copper sulfate, so it doesn't harm anything anymore. And then you need again copper sulfate instead of like thiourea in the last example, and HCl, which also neutralizes EDTA. So here you see how everything is like what you need to add into the test tubes. Here you see how it looks before the reaction, and then here how it looks after the reaction. So in the first Eppendorf tube, you add in every one of them you add water and the enzyme. And then in the first one, you don't add any copper sulfate. In the second one, you add 20 microliters, and in the third also 20 microliters. And as you can see here, in the first one where there is no copper sulfate, there is a color change. Why is there a color change in the first test tube? You can write your answers in the chat. Or you can also always unmute yourself if you want to share your answer with us. Okay. Exactly. This is the correct answer. It turns pink as there is no inhibitor or like no copper sulfate present, whereas in the test tube with the copper sulfate, which is the second one, as you can see here, it has copper sulfate in it. There is no color change at all. So this is a much more severe effect on the enzyme activity than we saw with the urea, which was a competitive inhibitor. This one kind of destroys the enzyme, at least for a certain amount of time. And now you see in the third test tube, at the beginning, it shows no color change. But then after 20 minutes of incubation, you can see it also gets red. And now if we look back at the table with all the ingredients added, you can see in the first two, in like with the one without color change, there is also copper sulfate present, which causes the enzyme not to work. But there is, in comparison to the when we looked at them before, here we also have EDTA present. And so what do you think is the effect of EDTA on copper sulfate and therefore on the enzyme activity? This is absolutely correct. The EDTA, when it's present, it is like it's chelating the copper ions, allowing the enzyme to remain unharmed and successfully turn the solution pink. Perfect answers. That's amazing. Okay. So this was the second activity. In the PDF that I shared with you, there are some more questions like about proteins and in more general, the catalytic effect of enzymes. I don't think we need to look at all of them right now, but you can maybe, if you later have time, you can look at the questions. However, what you can do now is, or maybe also later, if that works better for you, here I'll send a, the link to a form, and you can fill that form out. It has some questions about things we covered in today's webinar, and if you fill, oh wait, I I realized there's no. Oh, I have to make a change to the form, but um, maybe you can wait for filling it out as I have to add a section where you can add your name so you can get a certificate. Okay, here's the new link. And it's absolutely fine if you don't know all the answers. You can, for example, I'll stay in the call now, so you can ask any questions. Also, if you have um, if you have general questions, but also specifically to the questions inside the quiz. And you actually get a certificate of participation in this webinar in a few days. You just have to enter your email address, and by filling out the quiz, you'll receive an email with the certificate in a few days. There will also be a recording. There will also be a recording that we'll upload maybe tomorrow, and we'll share it in our Discord server. So you can, if you want to do the quiz later, you can always look back to the content. And obviously, using the internet, web search, Google, ChatGPT is also allowed, as you were really active in today's session, and I really enjoyed, yeah, having such engaged participants. So now is the time for asking questions. You can unmute yourself or write in the chat if you have any questions. Okay, of course. Wait, I have to find a good diagram about the inhibitors. Okay, I hope you can see my slides again. I'll also share the slides directly in the chat so you can access them. Here are the slides. Okay, so we'll go back to this slide. Wait, where is it? Here. As you can see, normally there's induced fit theory and the lock and key theory. Basically, what you need to know is that the substrate and the enzyme have pretty much the same shape, so they are like a lock and key. But there's also the more accurate version, which is the induced fit model, which states that the an enzyme and a substrate molecule is not exactly like the active site, but it is being stretched and put into this form, and by stretching the bonds, it is easier for the intermediate state to um like for the intermediate state that like to lower the activation energy. But as you can see here, it fits in. But now the competitive inhibitor, it blocks the active site. And we saw that with thiourea. It has a similar structure, but nothing can happen with it because it's not a real substrate molecule. And so when you add more real substrate, the effect gets less and less as kind of the real substrate outcompetes the competitive inhibitor. But with an allosteric inhibitor or a non-competitive inhibitor, it binds to a regulatory site of the enzyme. This is, for example, in hemoglobin and in other molecules, enzymes, and other proteins where the affinity of the substrate to the enzyme increases or decreases if an inhibitor or an activator, it can also be an activator, is present. I hope this diagram clarifies your question on the inhibitors. You can always ask follow-up questions. Okay, great. So are there any more questions? Yeah, you can submit it whenever you have time, and we'll also send it in our Discord server so people who m- couldn't join and still watch the recording can also fill it out. So no pressure. We'll send the certificates in about a week. Um, yeah, I hope you all stay engaged in with STEM2Connect, and there are more opportunities to come. We'll announce everything in our Discord server. I think there is a speaker event in somewhere in March with an expert giving a talk. And I hope you can join there then too. We'll announce everything in our Discord and also on LinkedIn and Instagram. You can get more updates.